Should I get started anyway? Okay, so I'm going to introduce myself, please, since it seems that we may have issue with the Zoom. Let me know if anyone can't hear me at the moment, and otherwise I will get started. So I am Marie-Dominique Filippi, in the Division of Experimental Hematology at Cincinnati Children's Hospital in Ohio, and I am currently serving as the director of the Cell and Procurement Core for the CCEH of Cincinnati Children. And in a sense, so the core that we have here is offering really three main areas of services. One is flow cytometry, the second is imaging, and the third is RNA-seq data analysis. And I think the presentation today will be focused on the science, but at the same time illustrate the type of services that here at Cincinnati we offer. And I would be happy to discuss if there are any interest in using those services. Okay, so my interest is really about understanding the regulation of hematopoietic stem cell function, and in particular stem cell self-renewal. And we've been doing that now really in the context of hematopoietic stem cell regeneration, either post myeloablative chemotherapy or post-transplantation, really for the goal of understanding how hematopoietic stem cells are able to reconstitute the entire blood system, and what are the regulatory pathways that are being used to do so. So here it is. Okay, so we all know how the hematopoietic system works, or we think we knew how the hematopoietic system worked. We know that the entire blood system is being generated from these long-term hematopoietic stem cells, which are mostly under quiescent state, and they're going to divide time to time to generate progeny, which then are going to commit to differentiation into all of these blood lineages and produce blood throughout life. Initially, for the longest time, we thought that how a stem cell makes mature blood cells was going through different differentiation stages. And that was happening in a stepwise manner. And at each steps, all of these population were thought to be relatively homogeneous entities. So there was a pool of stem cells, there was a pool of progenitors, and a pool of more committed progenitors, and so on and so forth. And I think now in recent years, we are discovering that the system is much more plastic than that. And instead of being, unified series of steps, it's mostly a more heterogeneous stem cell pool, which can take on many different ways to actually generate the same cells. And the reason why I'm pointing this is all of the knowledge that we've had so far on how the hematopoietic system is regulated is really using this stepwise model, and we are revisiting all of these steps, and there are many prior knowledge that we had that may no longer be applicable. And I think this is maybe the reason why the field has failed to be able to amplify stem cells either for ex vivo or even in vivo for the purpose of generating more stem cells for stem cell related therapy. Maybe because we don't completely actually understand what a stem cell is, how stem cell self-renewal is regulated, and whether stem cell self-renewal happens at all. And I will definitely get to that. So as I said, we are interested in how stem cell function is regulated during regeneration, really for the purpose of understanding clinical stem cell transplantation, because there are a number of limitations. And the main limitation in that clinical application is sometimes we don't have enough stem cells, and therefore the graft failed. So there is a need to expand these stem cells either for normal bone marrow transplant or during gene therapy. The second idea is that post-transplantation patients can sometimes remain at suboptimum hematopoietic blood count, and that could be or at higher risk of secondary neoplasm. And that could be because there are not enough stem cells which are being transplanted originally, or because the stem cells are sustaining stress and damages that make them functioning less. And so we really, our goal is to identify mechanisms of hematopoietic stem cell self-renewal to improve stem cell function under regenerative conditions. So for the longest time, we thought that actually hematopoietic stem cells had the ability to self-renew. And we are maybe discovering that it may not be the case. And this model, that I'm going to explain, is really the model that really points to the reality that true self-renewal may not completely exist, but instead, hematopoietic stem cell function may be lost each time a stem cell divide. So to be able to show that, researchers have used the H2B retention modeling system, in which that histone 2B can be transiently labeled because it is fused with GFP. And then we can stop the expression of H2B GFP and chase, and each time a cell divide, there is a dilution of GFP. So we can track hematopoietic stem cell division in vivo based on the dilution of these reporter H2B GFP. And while original work from Andreas Trump has shown that the pool of stem cells which was retaining H2B GFP labeling after 5 months of chase were the true repopulating cells, subsequent work from the Terry Moore group has shown that the GFP positive fraction can be subdivided in 3 or 4 populations, which we believe represent various stage of division, and each time a cell divide, it's actually losing a little bit of its function. So it's more a gradual loss of function rather than a population which would maintain self-renewal for X number of months and then lose it. And so we can easily model that using serial bone marrow transplantation in mouse, where each time we do 1 round of transplantation, and if we don't change the number of amount of donor cells, these donor cells are basically contributing less and less to producing blood each time at each round of repopulation. So there is actually a gradual functional decline of HSC function with cell division and post-transplantation, and which may indicate that there are pathways which are activated that are limiting HSC function and are preventing stem cell expansion. So we really want to identify those pathways and see if we can modulate them to improve stem cell function. So a lot of work has been done to trying to characterize what happens when stem cells are activated during the regenerative phase, and what happened in the post-regenerative phase when the system goes back to steady state. So during regeneration, there is stem cell activation, and there is a massive remodeling. There is a mitochondrial activation. There is high protein synthesis level. There is activation of a number of stress signaling pathways. And this is absolutely critical for the stem cell to exit their dormancy state during steady state condition. And then the stem cells are able to go back to quiescence. But certain pathways which were activated during the regenerative phase remain high. They do not go back to the original state. So for example, reactive oxygen species remain high, activation of a number of stress signaling pathways remain high, including P38 signaling pathway, TGF-beta signaling pathway. And these changes are responsible for a functional decline of the HSC pool post-regeneration. So we really want to understand what happens during active division and in the post-regenerative phase that impose sustained damage to the hematopoietic system. And so because of that, a number of experiments that we are doing implies that we are always going to compare stem cells which are quiescent versus activated going through division, what happens in the post-division phase, and what happens when these cells are reactivated again. And so one experiment that we did was to compare this journey, the gene expression profile of stem cells going through cell cycle, and doing so from naive pool of stem cells versus a pool of stem cells from a transplanted animal. So we take the stem cells.
I'm going to test. Can you hear me?
Yes.
Okay, perfect. I'm on my phone. Something's wrong with my computer.
Okay.
Sorry about that, Marie.
No problem. Thank you. And so we compare basically the stem cell pool before transplantation, after transplantation, and we ask the question, what are the difference in gene expression when the cells are quiescent versus when the cells are activated in cycle? And so this is a map, and we did single cell RNA-seq of differential gene expression of stem cells when they are quiescent, when they are activated into cycle, before transplantation and after transplantation. And so globally, what we have is a massive activation of cell cycle and metabolic pathway during the active phase, which is not very surprising. Post-transplantation, the stem cell pool is able to go back to quiescence. We know that from cell cycle studies, but at the gene expression level, we also can see that the cells are going back to a dormancy state. They are no longer active in cycle, and they no longer express genes that are necessary for metabolic activation. But the interesting part is if we try to reactivate these cells, what we noticed is the metabolic reprogramming that was happening during activation was no longer happening. So after stress, stem cells fail to reactivate again. And what we noticed is most of these genes belong to mitochondria or nuclear encoded mitochondrial genes, and therefore we decided to examine what happens to mitochondria during this cycle, quiescence activation, post-transplantation reactivation. So we know that mitochondria are the powerhouse of the cell. They are generating ATP for energy production, but it's much beyond that. They are serving various purposes, including for biosynthetic purposes, nucleotide synthesis, lipid synthesis, and they are also producing all the metabolite intermediates for epigenetic remodeling. So mitochondrial function are way beyond energy production. So if we look at a mitochondrial parameter, essentially mitochondrial membrane potential, MMPs, or mitochondrial reactive oxygen species, as a marker of mitochondrial activity, we can see that there is mitochondrial activation with activation into cycle. So for this specific experiment, we actually used poly-IC and inflammatory stress to activate the stem cells into cycle, and there is mitochondrial activation. We do see the same after 5-FU or after transplantation. But what happens when the system, after activation, when the system goes back to steady state, there is actually a loss of mitochondrial membrane potential. So the mitochondria are no longer activated, but they're actually sustaining damage and losing their mitochondrial membrane potential. And we see the same thing, whether it is post-transplantation stress or post-5-FU stress. Mitochondria are very dynamic organelles. They are all constantly going through fusion-fission cycle to generate a connected network versus to generate smaller unit. This fusion-fission cycle is important for quality control mechanisms. It's important, fission is important for mitophagy to degrade dysfunctional mitochondria. It's important for the distribution of mitochondria in the cell. It's important during cell division. Fission, on the other hand, is important for maximize mitochondrial activity and oxidative phosphorylation. So we decided to look at all of these parameters in our context. And so the first thing that we noticed using immunofluorescence microscopy, and these are 3D images, Z-stack, 3D reconstitution through Imaris software, one can see that in non-transplanted stem cells, the mitochondrial network in green is dispersed all around the cells, and we can see filamentous mitochondria or smaller unit, which is an indication of active dynamism. But after transplantation, the mitochondrial network is different, is much more compact. It's polarized in one part of the cell, and we can see less defined mitochondrial organization, if you will, in these cells. And if we use electron microscopy to look at that in greater detail, and this is a zoom image here we can see that mitochondria are round with defined cristae in stem cells before transplantation, but after transplantation, the mitochondria are more swollen, elongated, with less defined cristae. So there is sustained structural damage and damage in organization of the mitochondrial network after transplantation, which is not basically repaired, regardless, despite the fact that the stem cells are going back to quiescence. We used a different animal model to test whether the stem cells were maintaining damaged mitochondria because of a lack of mitophagy, so inability to remove damaged mitochondria. And we used the KIMA system. So the KIMA, it's a fluorescent protein, which is pH sensitive, which has been fused to a mitochondrial targeted element. So that reporter, fluorescence reporter, coats mitochondria, and when the mitochondria are in the lysosome, which would be indication of mitophagy, the pH drops and the fluorescence properties shifts. And one can see that as a shift from green to red. So green mitochondria represents the mitochondrial network, red signal represents mitochondria into lysosome, and we infer that as mitophagy. And so in a naive stem cells, we can actually separate cells which have high level of mitophagy, so very high red signals versus lower red signals, and we can appreciate here the difference. And the repopulation ability of the stem cell pool is really found in the stem cells which have the highest level of mitophagy. And if we look at that after transplantation, we lose the stem cell population which has high level of mitophagy. So we think based on these data that we can infer that there is loss of mitophagy activity in stem cells after transplantation, and that may be the reason why the stem cells are no longer able to get rid of their damaged mitochondria. So all of what I showed you, difference in structure, organization, mitophagy, could be due to impairment of mitochondrial dynamism and especially fission, because fission activity is necessary for mitophagy and organization of the mitochondrial network. And one fission regulator, it's called DRP1, and we can do immunostaining of DRP1, and DRP1 is going to localize onto the mitochondrial network and allow fission of filament. This is extracted from this paper, and that were live images to show that DRP1 here in green can separate to filament. And so we did static images. Our color code is the reverse here. Green are mitochondria, red is DRP1, and through deconvoluted images, 3D reconstitution in Imaris software, surface building to really analyze the fluorescent signal, we can see that DRP1 in red is localized as fission point between two mitochondrial units. And if we quantify the amount of DRP1 in stem cells after transplantation is significantly reduced compared to non-transplanted. So we think that there is loss of mitochondrial quality control mechanisms after transplantation, and because of that, mitochondria which were remodeled during the activated phase cannot go back to their original state, and stem cells cannot get rid of these damaged mitochondria because of lack of mitophagy. And we can, using a DRP1 knockout animal model in which we can mimic the lack of mitochondrial quality control. We can show that losing DRP1 function causes functional decline of the HSCs. One can see this in serial transplantation, and it's also inducing a reduction in mitochondrial membrane potential after transplantation, more so than in wild-type control. So what I've shown you so far is that hematopoietic stem cells cannot get rid of their abnormal mitochondria, and that's probably a causal factor to inducing a functional decline. And so now we are asking two main functions. What are the functional consequences on HSC function and metabolic activity of HSC? And then we are really asking that question to determine whether we can actually exploit this knowledge for clinical application. In another word, if we identify a metabolic pathway which is normally used by HSC but becomes abnormal after transplantation, can we restore metabolic activity of HSC and make better stem cells?
Can I ask a question?
Absolutely.
Can you hear me?
Yeah.
Do you know, so you showed less DRP1 at those fission sites, and you are referring to mitophagy. Do you know if their lysosomal function is intact?
So that's a very good question, and we don't know that yet. So this is something that we are actually working on at the moment, but we don't have data yet. It could be both. Okay. Honestly, it could be both. Yes, but thank you. This is a very good question, and this is definitely something that we are going to look at.
Okay, thanks.
So we ask these questions, but we decided to take a step back a little bit, because comparing stem cells before transplantation and after transplantation, it's almost like comparing apple and oranges. It's really comparing stem cells which are already exhausted. And therefore, if we want to prevent this exhaustion, we need to understand how exactly functional decline with division happens. And so for that, we decided then to use the H2B, sorry, I realize it is not labeled. This is the H2B reporter mouse system, in which the mice express, can express H2B, GFP, under a doxycycline inducible promoter. So we treat the mice with dogs, we induce the labeling, and then we stop the dogs, and therefore and we wait for 5 months to be able to identify different stem cell population with different division history. And this is what is illustrated here. GFP is in the x-axis, and we can identify cells with really high level of GFP, medium level of GFP, and no GFP. And so because we are talking about mitochondrial function and activity, we also looked at this from the perspective of their mitochondrial membrane potential using TMRE. And you can see here that we can almost identify 5 populations: stem cells which have different level of GFP, and stem cells which have different level of mitochondrial membrane potential. And this is the same, but it's just analyzed a little bit differently. And if we isolate, if we separate these different level of GFP here, that we can artificially label as division 1, 2, 3, 4, we really see that for each population, we can identify 2 peaks of mitochondrial membrane potential. Sorry, I don't know if you see the label. I thought I had gotten rid of that. Hide, yes. So you can see that there are actually 2 peaks of mitochondrial TMRE. So meaning that we may have multiple population here, GFP high, GFP medium, GFP low, and within each GFP peak, mitochondrial membrane potential low versus high. And based on many studies in the field, we know that mitochondrial membrane low versus high represent different level of HSC activation. And so we decided to examine these in a more detail. And so first, we decided to look at what is within each of these population, what is the expression of other stem cell markers. So for example, EPCR, which is highly expressed in stem cells, CD34, which should not be expressed in stem cells, or CD41, which is supposed to be a more age type of megakaryocytic type of HSC. And one can see, especially for EPCR, that the expression of EPCR is the highest in these G1 cells. And there is a gradual decline in EPCR expression from G1, G2, G3, G4. And the reverse for CD34. Between this population, there is a gradual increase in expression of CD34, CD41, there's not much changes, such that if we use t-SNE representation of this flow cytometry data, and if we include the parameter of division and mitochondrial membrane potential, we can almost resolve the heterogeneity of the stem cell pool. These can have many implications. Could be that we are basically isolating different subsets of HSC, or HSC that have different division history, different mitochondrial membrane potential, and are going to different stages in their activation or their metabolic status. And so we did isolate this population, did transplantation, and there was a lot of data, so I'll try to summarize the main conclusion of all of these transplantation data. And if we look at the level of blood chimerism from this donor population, we can see that the highest chimerism is given from these G1 cells and the lowest with G4 cells. These cells here, we also, the one which are really GFP negative, do not engraft at all. And these cells here, which have lower level of GFP, so higher amount of division, but same mitochondrial membrane potential, have a lower level of chimerism in transplant studies, and that can also be seen in their bone marrow, and really the population which is able to reconstitute the stem cell pool is really from these G1 cells. And if we do a secondary transplant, we do see also that these G3 cells give rise to a lower chimerism compared to G1, but also, and interestingly, actually, their graph may be low quantitatively, but it's myeloid biased. And this is typical from a more age type of stem cells, which are low input stem cells, but myeloid biased stem cells. So these data are telling us that a division is inducing a functional decline. We are confirming data from others, but we can also separate various hematopoietic stem cell population based on their division history and mitochondrial membrane potential. And so now, actually, we can compare stem cells which have the same mitochondrial membrane potential, but different division history, to really ask the question, what is the mechanisms behind these stemness loss upon division by comparing populations that are comparable? And we hope to identify the onset of events that induces HSC functional decline by comparing these two populations in order to make better stem cells. So we basically did a number of experiments to trying to understand these mechanisms. And for that, we really focused on comparing these two populations, G1 versus G3, for less division, low mitochondrial membrane potential, high division, low mitochondrial membrane potential. And so the first experiment that we did was, sorry, I'm going back, is we wanted to understand a little bit the this myeloid bias was striking. So what we did was we transplanted these cells into animals, took the bone marrow of these transplanted animals from this population and did single cell RNA-seq. And from these data, we can see that, and this is a UMAP representation of the single cell RNA-seq of these bone marrow reconstituted by G1 cells versus G3 cells. The relative proportion of these various hematopoietic cells is different. So we did these experiments in collaboration with Lee Grimes and Nathan. These experiments were done by Annie, who is a student of Lee Grimes. And the nomenclature is the Lee Grimes nomenclature, who has said that there are a variety of stages in myeloid differentiation, neutrophil differentiation, from these pro-new, pre-new, immature neutrophils. We see here monocytes, the cluster of monocytes, and here the cluster, I should point that here the cluster of stem cells with multipotent progenitors. And one can see the classical separation of the branching of the lineages from HSC. We see the branching to megakaryocyte and erythroid population versus the myeloid one. And then the B cells are completely separated. So what we can see is the marrow reconstituted by G3 is quantitatively different than G1. The relative proportion of all of these various steps of the differentiation process are different. There is an enrichment of this immature neutrophil. There is a loss of these pre-new, pro-new cells. There may be an enrichment of the monocytic population and a decrease in the stem cell pool. And if we zoom in to these HSC progenitor population, so the HSC are on the top. Here there is the branching of the megakaryocytes and the erythroid progenitors, the MPP4, the multilene, and so on. If we compare from non-transplanted cells, transplanted G1 versus transplanted G3, transplanted G1 are fairly similar to the control in terms of the relative proportion of all of these various stages. However, G3 again is completely different. We see that the HSC, which are reconstituted by G3, are at the top of this hierarchy, but there is a decrease in intermediate progenitor, and there is an abnormal ratio of erythroid progenitor to megakaryocytic progenitor compared to G1. So really, the repopulation of the bone marrow is qualitatively different when it is reconstituted from HSC, which low division history versus high division history. So G3 has a low input, but also the trajectory of the differentiation is different. And I think this is an important result, and that we need to understand further why the repopulation pattern of the hematopoietic system is different. The second thing from this data that we were not anticipating at all is there is also in each of these population differential gene expression. So these are the cells which were derived from G1 and G3. And not only the trajectory of reconstitution is different, but from each of these populations, there are differentially expressed genes, and which means that the mature cells are retaining a signature of a stem cell which has divided. So we are going to call that the division memory, which is being transmitted to the progeny. And what are these differentially expressed genes? So we have division genes, so that may be the division memory. We have genes which belongs to spliceosomal complex. We haven't looked at those yet, but we definitely need to do that. And then we have a huge signature of nuclear encoded mitochondrial genes. So again, we know that there is damage to mitochondria after division, and not only the stem cells do retain the memory of that damage, but it looks like they are also, that probably impair their ability to produce blood, but also this memory is being transmitted to the progeny. So something we haven't done yet is really to try to see if these more mature cells are functionally different or not. It's something we should be doing. And so for the last 5 minutes, so we know that now these data that are telling us that the blood which is being formed by these stem cells, which are different by their division history, is different. So what is the mechanisms? And so for this, we isolated these cells, and we did the bulk RNA-seq of comparing these population, but we did that when the cells are actually actively cycling. Again, with the idea that we are really looking at mechanisms that are division dependent. So really on purpose, we decided to activate these cells to determine what are the regulatory pathway that these cells are using to actually make blood. And so this is what that heat map represents. We did this in Nathan Salimoni's software alt analyze, and this is just an unbiased hierarchical clustering of these of the differentially expressed genes of each of these population. And so the first thing is these cells are different. When you activate these cells, they do not express the same the same genes. And I was personally not anticipating that the difference would be that important. And so mining these data, we can identify a number of differences, but one more prominent difference is about cell cycle and about differentiation. So these cells seems that they are not, once you activate them, they are going to be primed different way. So these cells are not yet differentiated, right? We just activate them for 24 hours. But some of these cells start expressing lineage markers, and more so this G4 population, for example, that G1. So the kinetic of priming is completely different. And it's not just any type of genes. These G4 actually cells are seemingly, or at least their gene expression is telling us that they may be mega biased. It's something that we are going to have to examine. But you can see that none of them express the same amount of priming gene signature. And so because of that, and also it looks like they are not in the same phase of the cell cycle after 24 hours in culture. So we examine cell division kinetic of G1 versus G3. And you can see here, this is a graph of accumulating cell division over time, that G3 is cycling faster than G1. And these are the subsequent second and third division. In this, if we ask the question, and this is a single cell assay, and this is a single cell differentiation assay, when we let single cell to produce a colony and we examine whether that colony is multipotent or not, G1 is giving rise to almost 100% multipotent clones, but there is significant reduction in, although it's still high, of multipotent clone produced by G3. So these cells do not cycle the same speed, and they are not going to be lineage primed the same way. And that could indicate that they are not going to differentiate the same manner, which would be consistent with the single cell RNA-seq data from the bone marrow that these cells have generated. And we still need to understand the mechanisms behind that. But for now, we are going to focus on what could explain the difference in cell cycle progression, and could it be associated with a loss of stem cell function. So the second signature that we found, which was different across this population, was the expression of metabolic genes. And unfortunately, I don't have time to go into too much detail because there are a lot of information behind this, but one of these pathways, which was more highly expressed in these G1 cells, which are the most stemness cells compared to G3, was branch-chain amino acid catabolism. And so if we just express these data as network, one can see that there are a lot of metabolic pathways. So branch-chain amino acid catabolism is high in G1 cells, so the one which have long-term HST activity, and the G3 cells have more a signature of being myeloid biased. And so we decided to focus on these branch-chain amino acid catabolisms. So because branch-chain amino acid catabolisms, including BCAT2, which is the an enzyme of the initial step of branch-chain amino acid catabolism, happens in mitochondria. And BCAT2 was more expressed in G1 compared to G3. So branch-chain amino acid are leucine, isoleucine, and valine. And so these amino acids, they can enter mitochondria, and instead of being used in the cytoplasm to make protein, they go into mitochondria through this transporter and are being catabolized instead. And they're forming a variety of metabolites intermediates, and each metabolite intermediate is being used in a different way. Some are going to form acetyl-CoA, succinyl-CoA, which can enter the CCEH cycle. Sorry, acetyl-CoA can be used to generate lipids, or downstream of that metabolite intermediates, KIC, it can go towards cholesterol synthesis. So to determine whether this pathway was important for stem cells, and to assess whether loss of stem cell function could be attributed to a loss of BCAT activity, we use two approaches. One, to use a BCAT inhibitor and to inhibit this in fully functioning stem cells. And the second is to use KIC to supplement stem cells after a division, which is less functioning, to see if we could restore its function. And we have a proof of concept that indeed these branched-chain amino acid catabolisms could be a reason why cells after division are less functioning. So if we inhibit BCAT with the pharmacological approach in fully functioning stem cells, we can create a more myeloid biased graft, at least in the bone marrow, compared to DMSO control treated cells. We can accelerate the cell division of this population, and we can lower the multipotent, the potential at the single cell level. So we are basically, if we inhibit BCAT activity, we can mimic a certain feature of G3. And then the other way around is we took these cells, G1 and G3 essentially, and asked the question whether supplementing G3 with metabolites from branched-chain amino acid catabolism, could we change the biology of G3? And so if we really focus here, if we treat, sorry, this is a mistake, it should be G3. If we treat G3 with KIC, which is a metabolite produced by branched-chain amino acid catabolisms, we can slow down their cell cycle. We can prevent their activation, and we use phospho-S6 as a marker of activation here. And if we use CD48 as a marker to see how fast these cells are going into differentiation, this being G3 compared to G1, which acquire in culture CD48 faster than G1, we can suppress that as well by incubating the cells with KIC. So these data are telling us that, and we don't have the full set of data with engraftment data, but it's going to be, I think, very promising, is the fact of going through cell division, alter mitochondrial function. It does have, that are not repaired. So the cell never recover from that. It does affect their subsequent metabolic activity and use of metabolic pathway for their function. And I think this is really an essential driver of losing stem cell function. And so we really need to integrate as a whole transcriptome data, metabolomic data, to really identify what pathway are being used by hematopoietic stem cells and which pathway are being deregulated after stress of regeneration. And I believe that that could be used as a precision mitochondrial metabolisms as a therapeutic approach to maintain better HSC function either in vivo or ex vivo. And I'm really looking forward to pursuing that work in this regard. So I'll finish by thanking people in the lab who have done these experiments. The first part of the talk was basically done by former postdoc in the lab, Ashwini Hinge and Jihi Ki. And the second part has been essentially done by a talented graduate student in the lab, James Bartram, who is, by the way, going to graduate very soon. And is looking for a postdoc, and had help with a member of the lab. lab. And a big thank to our collaborators, especially Lee and Nathan, for the analysis of the single cell RNA-seq. Thank you, and I'm happy to take any questions.
Great. Thank you, Marie. Appreciate that. Sorry, everyone, for the problem with my audio. I still am not fixed. I'm speaking through my phone. So any questions that anybody wants to ask, go ahead and unmute yourself and ask. I don't see any coming in the chat so far.
I don't see any questions either, which is perfectly fine.
I have a quick question, Diane.
Okay, go for it, Aki.
So in a recent blood paper, there's the self-renewal factor, ANGPTL2, has been described as the driver of the self-renewal. And then I just looked at the ANGPTL2 signal transduction pathway, that whether that's overlap with the mitochondrial respiration function and fission and fusion. And there are some indications that the this ANGPTL2 generates the signal that affects a mitochondrial function, I guess. Do you have any evidence of this, the self-renewal factor might driving a G1 to G3 potentially after the transplant?
So this is a very good question, and I absolutely need to read that paper. Actually, I really believe that what's wrong with division is the metabolisms of HSCs, and that could imply many regulatory elements of mitochondrial function, and we're definitely going to look at that paper in particular and that gene. It seems very interesting. Thank you.
Yeah, Aki, if you have that reference, you could populate it in the chat.
In the chat, that would be fantastic.
Okay, I will.
Thank you.
Thank you so much. Other questions?
Yeah, this is Stephanie. Hi, this is really, really, really awesome, and my brain is blown. So much fantastic information. So can you, and I'm apologize if I missed that, but can you get at it in human cells?
So we are doing those experiments as we speak. And so we are literally going to look at that from bone marrow samples from patients to see if we can find differences. Very early, I can perhaps still tell you that treating human CD34 with the BCAT inhibitor, we are seeing that it is impairing hematopoietic stem cell function. So it's promising that it's a conserved pathway, and we are going to see the same thing in human cell as well. But yes, we're definitely doing those experiments, and I'm hoping to even move in a disease model as well. But need more funding for that.
Always.
Hint, hint, hint.
Exactly.
Any other questions? If you're on study section, now you've just heard the talk and you should support the grant.
Oh, thank you. Well, that will be submitted soon. Stay put.
We have a question in the chat from Yawin. Do you want to ask it, or do you want me to read it?
Oh, yes. So the question is, could you explain how you prime G1 and G3 cells into cell cycle? So we are using culture conditions and SCF-TPO to push to cell cycle, which are very canonical way to activate stem cell ex vivo. AKT, we haven't looked at AKT in G1 and G3. If I remember correctly, we did look at AKT when we did some experiment comparing non-transplanted and transplanted stem cells, and we didn't find a difference. We have difference in mTOR activity. have a difference in ERK, but I don't remember if we had a difference in AKT. But it's something that we need to look into more detail, and we'll definitely do that. Thank you.
Great.
And thank you for the link. Definitely look at that paper. Yeah.
Can I ask one more quick question?
Oh, absolutely. Please go ahead.
So for human, to mobilize the HSC, we use the GM-CSF. Now, the obvious question is, does GM-CSF treatment and mobilized stem cells in the circulation getting more toward a G2, G3 rather than G0, or?
So mobilized HSC are deeply quiescent, and it could be that mobilization. I think Terry Moore had a paper actually on that, trying to assess using the H2B model which population was being mobilized. Okay. And I honestly do not remember exactly the details, so I may be wrong, but it may mobilize preferentially a G1 population, winning more STEMI population than not. But I think she had a blood advance paper on that.
Okay, thank you.
Thank you. And thank you everyone for logging in this afternoon.
Marie, I just want to apologize again for my audio.
Oh, no worries.
I didn't get to introduce you or anything. So thank you so much for your willingness to speak.
No worries about that. Thank you, Diane.
Okay, everyone take care, and we'll see you next time.
Exactly. Bye.
Bye.